World, we've heard a lot of terminology thrown about. And even those of us with strong scientific backgrounds can still be perplexed by words we know but don't truly understand the science behind. Well, today we're in store for some amazing insight into the hyperinflammation syndrome referred to as cytokine storm, or CSS. This disorder did not just begin with SARS-CoV-2 infections, but has been studied for quite a while and has been thrust into the spotlight with coronavirus infections, as a subset of patients experience the ill effects of this overreaction by the immune system. In our presentation today, we'll learn about the cellular and systemic conditions that regulate CSS and its association with COVID-19 and the innate immune response. Now it's my pleasure to introduce you to our esteemed speakers for this presentation. Dr. Randy Cron is Professor of Pediatrics and Medicine and Director of the Division of Pediatric Rheumatology at the Children's Hospital of Alabama and the University of Alabama at Birmingham. Dr. Cron's laboratory research focuses on the transcriptional regulation in CD4 T lymphocytes as it relates to both systemic lupus erythematosus and HIV-1 AIDS. His clinical and translational research interests include childhood arthritis-related macrophage activation syndrome and temporomandibular joint arthritis, for both of which he is known worldwide. Dr. Cron is also an expert in cytokine storm syndrome and will discuss in detail his findings on the disorder. Dr. Frank van der Veerdonk is a professor at Radboudumc University Medical Center for Infectious Disease. Dr. van der Veerdonk is an internist and infectious disease specialist that cares for patients with fungal infections and primary immunodeficiencies. His research has focused on chronic mucocutaneous candidiasis, hyper-IgE syndrome, and chronic granulomatosis disease. He is currently coordinating clinical trials for immunotherapy and fungal infections and has a specific interest in influenza-associated aspergillosis. In his presentation today, he will tell us about some unique pathology he has been seen associated with COVID-19 cases and the immune system role in its development. But before we begin, I want to remind the audience to stick around for the Q&A session that will occur right after the last talk. Don't be shy. Send in your questions at any time. Any question you have at all for our panelists. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. Now let's get to why we're all here. Randy, I'm going to turn the mic over to you.
Hello, my name is Randy Cron. I am a pediatric rheumatologist and professor of pediatrics at the University of Alabama at Birmingham. And we're going to talk today about cytokine storm syndrome, kill or be killed. These are my disclosures relevant to this talk. So cytokine storm syndrome is an umbrella term under which there are a variety of conditions that are all related by a hyper-inflammatory cytokine state, such as macrophage activation syndrome, as termed by the rheumatologists, the familial or genetic form of hemophagocytic lymphohistiocytosis, or HLH, cancer-related cytokine storms, such as leukemias and lymphomas, and cytokine storms or cytokine release syndrome that occurs in treatment for refractory leukemia, such as CAR T-cell therapy. And there's this large Venn diagram circle of infection-triggered cytokine storms, including many viral triggers, such as bad strains of influenza, Epstein-Barr virus, and other members of the herpes virus family. And now we have COVID-19. Acute respiratory distress syndrome can certainly be part of cytokine storms, but can also occur outside of cytokine. So my interest started with this 14-year-old girl who came with a prior diagnosis of cytophagic histiocytic paniculitis, and she'd had fevers every single day of her life and total body paniculitis, or inflammation of her fat. And she was the size of a 7-year-old, probably from chronic steroid use and the chronic inflammation. And seeing the word paniculitis, we treated her with cyclosporin and corticosteroids, and she got a little bit better, but a month later she was back in the hospital, this time seizing. She had belly pain. She quickly became comatose. She had inflammation in her cerebral spinal fluid. She had coagulopathic features. All her cell lines were down. She had this elevated serum ferritin, a marker of cytokine storms, pro-inflammatory cytokine storms in her blood. and multi-organ failure, heart failure, lung failure, kidney failure, liver failure, pancreatitis, marrow failure, bleeding from all our orifices, and a bad central nervous system. This was a bone marrow biopsy showing these activated histiocytes or macrophages engulfing other cell types, or hemophagocytosis, kind of a hallmark feature of cytokine storm syndromes, but it's only present about 60% of the time. This is her fat from around 9 months of age, showing the fat cells, but infiltrated by these lymphocytes, or the paniculitis. There are criteria for cytokine storm syndromes, including familial hemophagocytic lymphohistiocytosis, or HLH. You can either have a molecular diagnosis or 2 gene mutations in perforin or perforin-related proteins in that pathway. Most of these are autosomal recessive disorders. Most people don't show up with this at birth unless they've had a sibling with it. And then there is these criteria which our patient met in spades. However, for secondary forms, they're often too restrictive. But she did have fever and a large spleen. Her cell lines were down. She consumed fibrinogen as part of the coagulopathic process. She had hemophagocytosis, as we showed you, in her bone marrow. She actually had low or absent NK cell function, hyperferritinemia, and elevated IL-2 receptor alpha chain or soluble CD25 coming from her lymphocytes. For me, she was one of the sickest kids I've ever seen, who essentially came out of the ICU unscathed. She was initially seen by our oncology colleagues, who started her on the HLH protocol, which is based on etoposide or chemotherapy, and she wasn't improving. And around this time, the end of 2004, we had started using a new drug, anakinra, which is a recombinant human protein that's an interleukin-1 receptor antagonist. And we had been using it in children with another condition, systemic juvenile arthritis, who would often get cytokine storms, and anakinra seemed to help them. This girl was going to die, and so we talked to the family, talked with the other team members, and decided to initiate anakinra for a cytokine storm, the first time this had ever been done. And within 2 days, she woke up from her coma. Within 6 days, she was out of the intensive care unit, and within 2 weeks, she was out of the hospital. It was really nothing short of miraculous. So what's the pathophysiology behind these conditions? So if we just take a generic strength of the immune response on the y-axis and time along the x, and let's even just take cytokine storm syndrome out of the equation for a moment. If you're a normal, healthy individual and you're moving along here in your daily life, and let's say you get an infection, for example, influenza, your immune system will mount a response to that infection. Hopefully it will start to clear that infection, and then your immune system is actually hardwired to shut itself down. Otherwise, we'd be one giant state of inflammation all the time. But what's thought to occur at cytokine storms like HLH is this immune system just keeps ramping up and getting out of control. And the thought is you have these cytotoxic CD8 T lymphocytes or natural killer cells that kill infected antigen-presenting cells through the perforin pathway. And if they're incapable of killing through that pathway, they remain engaged longer together, and they crosstalk and spew out these pro-inflammatory cytokines, leading to the multi-organ failure. And this was modeled very nicely by Michael Jordan, the pediatric oncologist, not the basketball player, when he was in Pippa Merrick's lab at the University of Colorado. And what Michael did is he took wild type or normal mice, or mice that were homozygous deficient in the protein perforin, and these mice do perfectly fine in the mouse colony until they get the wrong infection, in this case a virus called LCMV. Now the wild type mice, when they're infected, will produce pro-inflammatory cytokines like gamma interferon, and through perforin kill these infected antigen-presenting cells and just shut down the viral life cycle, and they do fine. Whereas the perforin-deficient mice will generate gamma interferon but are incapable of killing these infected antigen-presenting cells because they lack perforin. And these mice will uniformly die within 10 to 14 days. But the interesting thing that Michael did is he blocked interferon gamma with an antibody, and these mice had trouble clearing the infection, but they didn't die. Similarly, if he blocked the CD8 T cell population, they had trouble clearing the infection but didn't die. And this is a really important concept, which I think translates to humans as well, is that the virus is the trigger of this immune response, but it's the immune system that's killing the host. So there are a variety of genes, and this is a short list. It's getting longer every day. including perforin and other proteins that are involved in the perforin pathway. And if you're homozygous deficient in this, you will get a familial form of this disease. And this is modeled here in this cartoon, and we have either a cytolytic CD8 T cell or a natural killer cell. They share the same perforin pathway, and when it engages its target cell or antigen-presenting cell, it gets activated. And perforin, which is already pre-existing in these cytolytic granules along with granzyme, traffics along the actin cytoskeleton to this immunologic synapse between the two cells. And all these funny named proteins, Rab27A, Syntaxin 11, Monk13-4, for example, are involved in the trafficking, docking, or fusion of these cytolytic granules to this membrane here. Perforin is released into the synapse and actually forms a channel or pore between the two cells, allowing for granzyme to be delivered, which triggers this cell to die by apoptosis. And if you're deficient in any of these proteins in this pathway, you will get a cytokine storm at some point if this is triggered. And so when I arrived from Philadelphia in Birmingham about 13 years ago, we started getting consulted on patients in the hospital with cytokine storm. And some of these were patients that were our own patients with systemic JA, but many were not. And we were surprised to see that a high number of these patients, a high percentage of them, had heterozygous or single copy mutations, sometimes multiple mutations or multiple genes in these same perforin pathway genes, despite the fact they're not showing up necessarily in infancy when the familial patients show up. And so we hypothesized that even having a single copy bad gene in this pathway may contribute to decreased cytolytic function, and we used NK cells to study this, and therefore contributed to the cytokine storm. So the work I'm going to show you is primarily done by Mingse Zhang in the lab, and what Mingse did was make copy DNA using a human natural killer cell line that had all the perforin pathway genes intact, and he would make copy DNA of the wild type gene, and then by site-directed mutagenesis introduce the patient mutation. Then he generated lentiviral expression vectors for introducing the gene into the NK cells, and he would transduce these cells to overexpress more of the wild type gene that they already expressed, or the patient mutation. And then we studied natural killer cell function in a variety of ways. And so here's the lentiviral expression construct driven by the immediate early CMV promoter, which works well on lymphocytes, the cDNA for the gene of interest, and we'll focus on Rab27A in a moment here, either the wild-type gene or the patient mutation, this linker, so we could also co-express green fluorescent protein to track and sort these cells. And so I'm going to focus a large part of this talk on this 18-year-old who was previously healthy and had a mutation in Rab27A when she, and this is, most of these mutations are amino acid change or missense mutations, so changing one amino acid to the other. She was previously healthy. She was a competitive barrel racer, and she presented with 2 weeks of fever and belly pain. When I was called to see her, she had a fever. Her central nervous system wasn't quite right. You could talk to her, but she didn't make sense. She had a big liver and spleen, and they were wheeling her to the ICU when I met her. And her labs were very compelling. So all her cell lines were down. She had inflammation of her liver. She was coagulopathic. Her ferritin was up. All these underlying ones are markers of cytokine storm syndrome that are part of the HLH criteria. And we had developed a cocktail therapy of cyclosporin, high-dose corticosteroids, and this IL-1 blocker, anakinra. And clinically she improved, as did her ferritin levels normalized, her liver enzymes normalized within a few days, and her platelet counts returned to normal. So we took her mutation and we introduced them into NK cells, and we'll focus on this middle panel here, which is a summary of over 12 experiments, where we're looking at the ability to kill through the perforin pathway on the y-axis and increasing the effector to target cell ratio. So the more NK cells per target, the more killing you get. And if you introduce that empty lentiviral vector, you can see nice killing here. These are the circles. If you introduce more of the wild-type Rab27A gene, these NK cells can kill a little better. And if you introduce the patient mutation, they kill slightly less well. We confirm this by a degranulation assay. So when these NK cells get activated as part of moving the cytolytic granules to the immunologic synapse, they also spit out a protein CD107A onto the surface of the cell that can be detected by flow cytometry. So here is overexpressing the wild-type Rab27A, and over time, you can see kind of maximal expression of CD107A at about 2 hours, whereas the patient mutation, the degranulation was always less, and kind of summarized here from multiple experiments. So a colleague of mine, Mark Walter, is expert at in silico kind of modeling patient mutations, and here's the Rab27A crystal structure, and here's the patient mutation here in purple, and it was predicted to disrupt interaction with one of its binding partners, MUC13.4. It did that first by Western blots, where we did co-immunoprecipitation of wild-type MUC13.4 with either wild-type Rab27A or the patient's mutated version in the NK cells. And I think you can see at every time point, these are all controls down here, that the patient's mutation in Rab27A bound less well to wild-type MUC13.4 than wild-type Rab27A at each time point. We do that by a proximity ligation assay, which allows you to identify where two proteins in situ, inside a cell, are close enough to be detected by this assay, i.e. they're close enough to be interacting. And if you overexpress the wild-type Rab27A, every orange dot is being picked up by this assay where it's interacting with MUC13.4. If you overexpress the patient mutation, you have fewer dots, and that's summarized here. So this mutation does probably partially disrupt the ability of Rab27A to interact with its binding partner MUC13.4. If you look by confocal microscopy at this process, so E, or effector cells, these are your natural killer cells. Here's their natural K562 target cell. When they interact, all these granzyme is detected by yellow or green staining here, is polarizing to the immunologic synapse between the effector and the target cell. Some of it is not, but most of it is. And that's the empty vector control. If you overexpress more of the wild-type Rab27A, you get almost complete polarization, whereas if you express the Rab27A mutation, you get some polarization, but a lot of the granzyme B is not polarized in these cells. Now, this is a snapshot in time. So if we look at a time course from multiple experiments, multiple conditions, and then count 200 cells per slide. This is an immense amount of work that Minksa did, and he did it in a blinded fashion, so he didn't know which condition or time point he was looking at. You could see whether the Rab27A wild type had more polarized cells or the patient mutation, and here from many experiments. And so on the y-axis, we have percent of polarized cells at a population level, and time along the x. And so if you introduce the empty vector into these NK cells kind of maximally at a population level polarize granzyme around 1 to 2 hours. If you overexpress more of the wild-type Rab27A, that's a little better. But the patient mutation, it's about 2 hours delayed at a population level. Around this time, this really lovely paper came out of Australia, Misty Jenkins and colleagues, and they took CD8 human T cells that were deficient in perforin, or granzyme B, and they looked by time-lapse photography at what happened. And if they were incapable of killing their target cell, they spent 5 times longer engaged than the wild type, and that's shown here, so increased dwelling time. And as part of that prolonged interaction, they spewed out increased pro-inflammatory cytokines like interferon gamma and TNF. And so Minksa tested that in our system by looking at interferon gamma expression, by overexpressing either the wild type or the mutant, and it's summarized over here. So there's a percentage of cells expressing gamma interferon. The empty vector or overexpressed wild type expressed this amount of gamma interferon over time, but the patient mutation, which had delayed cytolytic granule polarization, these cells produced more interferon gamma. Now, I presented this at a meeting, and one of my colleagues from Rome said, we have a 14-year-old patient with the exact same Rab27A mutation, And he and his father, as did our patient's father, had that mutation, and that boy developed a cytokine storm. And both when he and his father were well, either by degranulation comparing to the sister and the mother who did not have the mutation, or comparing to healthy donors, their natural killer cell function was about half normal at baseline, which for the parents, the fathers, was fine because they didn't get the wrong trigger. And so we concluded that at least for this one mutation that we studied in detail in RAB27A, that it likely decreased NK cell function by a partial dominant negative fashion, contributing to the cytokine storm. So this paper came out in 2010, and it caught my eye. It was about the 2009 H1N1 influenza pandemic, which killed a fair amount of people around the world, including previously healthy individuals in the United States. And they reported on a subset of patients at the University of Michigan Hospital who died from influenza that year, and they had hemophagocytosis all over their pathology. We brokered a deal between our colleagues in Michigan and our colleagues in Cincinnati who were doing the genetics for us at the time, and 36%, or 5 of the 14 samples that we analyzed, had mutations in one of these perforin pathway genes list. 3 patients had this same mutation, and 2 of these 5 also had a heterozygous mutation in perforin 1. That this perforin 1 mutation, compared to the wild type, had decreased lytic activity. Again, suggesting that these, we all get exposed, or many of us get exposed to these viruses. Many of us contract the virus, but only a small percentage of people require hospitalization or die from the virus, and maybe it's because they have a baseline defect in their NK cell function. So our colleagues in Cincinnati who knew which genes to look for had this patient with systemic GA who got recurrent cytokine storms, but they could never find a mutation. And when they switched from whole exome to whole genome sequencing, they found in this first intron here of MUNC13.4, this blue line here, mutation, that was predicted to disrupt an NF-kappa B binding site that leads to an alternative transcript that's expressed by the lymphocytes. It starts at exon 2. And Grant showed nicely that the patient had roughly normal levels compared to control of the conventional transcript, but the lymphocyte transcript was about half normal, suggesting this mutation may have disrupted transcriptional or enhancer activity. So Minxa showed by gel shift that indeed a canonical NF-kappa B probe does indeed bind the P50 and P65 NF-kappa B components, as shown by the super shift. And if you compete with self, you can compete away these bands for the most part. The wild type sequence of this intronic NF-kappa B site competes pretty well, but the mutation competes less well, suggesting that it doesn't bind NF-kappa B as well. And so Minxa generated multiple reporter constructs to test for enhancer activity. And the bottom line is that this region does function as a transcriptional enhancer when either driven by the immediate early CMV promoter or the HIV promoter, and the patient mutation disrupts some of that enhancer activity. So who cares, I guess, if you have less expression of UNC13D? What does that mean? UNC13.4, UNC13D is the same thing. So we drove down expression of UNC13.4, UNC13D with siRNA at different concentrations, and we got a correlative drop in degranulation or NK cell function. So this is a hypomorphic effect, so not having enough UNC13.4 may contribute to cytokine storm as well. And this is modeled here in this threshold model of disease. So if you're homozygous deficient in any of these perforin pathway genes, including perforin, you are already at that threshold. These infants present usually within the first days, weeks, or months of life with the cytokine storm. But maybe you're walking around fine throughout your life with a heterozygous defect, which partially disrupts your NK cell function or your CD8 T cell function, and then you get a bad organism like H5N1 influenza, or in other parts of the world, leishmaniasis, that pushes you over this threshold, so you get cytokine storm. Or you're a patient with lupus or systemic GIA, and you're inflamed, and you have a heterozygous mutation, and, for example, Epstein-Barr virus pushes you over the edge. So we've been doing a retrospective case series of patients 44 patients that we treated at Children's of Alabama who had cytokine storms with this IL-1 blocker anakinra. The mortality is still pretty high despite its benefit. As part of this study, we did sequencing of these perforin pathway genes, including this one STXBP2, which is important for fusion. And if you had an STXBP2 heterozygous mutation, those patients did poorly. So we studied one of these patients who had an STXBP2, or also known as MUNC18-2. She had a heterozygous mutation. And by killing assay, and we're showing the dead cells here in the upper right-hand corner, increasing the effector to target cell ratio as we go down, overexpressing wild-type STXB2 and NK2 cells. You got nice killing here, but the patient's mutation at every effector to target cell ratio, 28 to 18, 32 to 23, was less. Our colleagues in Pennsylvania actually found a complete dominant negative set of mutations in a couple different families in STXB2, kind of confirming what we saw in our patient. And now we're doing a clinical trial to look at the ability of anakinra to help both children and adults with cytokine storm syndromes. And as part of that, the first 5 patients we enrolled all had, including multiple often, heterozygous mutations in these same perforin pathway genes. This was really quite surprising. And as we've gotten more patients, so 6 out of 9 now, or 66% of these patients had mutations in the perforin pathway, and then we identified 2 patients who had a mutation in this gene DOC8. And DOC8 is this GTPase that interacts with CDC42, which is now another known HLH gene. And in addition to those 2 patients, we identified 2 other patients who had DOC8 mutations. 3 of them were amino acid changes. One was a splice acceptor site variant. And by using CRISPR-Cas, we knocked out the NK cell lines. And if you completely knock it out, these cells can still kill. So it's not completely required for killing, but it does definitely disrupt it at the 2 different time points. If we introduce back the patient mutation, though, it partially restores, but does not completely restore the decreased killing. Well, we did it by, we couldn't do this by a lentivirus because the cDNA for DOC8 is too big, so we used a foamy-virus approach. And the summarized here, NK cell function when you introduce the patient mutation is decreased compared to wild type, as is degranulation compared to wild type. And the consequence of that is you get increased pro-inflammatory cytokine production, whether it's TNF or interferon gamma. To explore the splice acceptor site variant, we generated this construct with a green fluorescent protein that we cut in half and introduced the intron with either the wild type sequence or the patient mutation at the acceptor site. If you introduce the wild type sequence and transduce this into NK cells, you get nice expression here compared to this plasmid alone. It's actually slightly better. But the patient acceptor site mutation gives you essentially no expression. We confirm that by conventional exon trapping. Here's the patient mutation, you get no expression. So we now think that DOC8 is another gene that fits into this pathway of perforin pathway cytolytic activity contributing to cytokine storms. There are a whole variety of other pathways we now know that can get you there as well. And I'm happy to take any questions.
Wonderful presentation, Randy. An amazing body of work and data. We really appreciate you sharing it with us. Very quickly, before we move on, I want to remind everyone that the Q&A is going to begin right after this next talk. So send in your questions for both Randy and Frank. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. Okay, Frank, we're looking forward to your presentation as well, and the floor is now yours.
Thank you for the opportunity to present some of our novel data regarding COVID-19 and related to the inflammatory syndrome and dysregulation of COVID-19. I would like to present some topics, cytokine and complement signatures that we found in COVID and pathway analysis in moderate, severe, IL-1 biology, autoinflammation, and the novel kallikrein-kinin system being involved in COVID, and finally, treatment strategies and timing of COVID. When the COVID pandemic hit the Netherlands, we immediately put in a collaboration with the region to collect samples and plasma, and these are the measurements of cytokines in those plasma of patients of moderate and severe, basically admitted to the ward or directly to the ICU, and followed over time. The first signature that we saw was IL-6 being elevated in the more severe COVIDs, and along the way, after admission, this would gradually decrease a little bit but remain high. In contrast, TNF, we didn't find a real signature, not the elevation that we know from sepsis, and there was not much dynamics, not in the severe and not in the moderate. Finally, we also were interested in the complement system. We measured the terminal complement complex as a readout. Although it was increased in COVID-19 compared to sepsis, it was still relatively low. So there is an activation, but not that striking. We continued by doing protein analysis in the plasma of those patients. And here we have a presentation of patients that are severe versus the moderate, so the patients that are admitted to the ICU compared to the patients that are more admitted to the ward. Here you can see that when it's on the right, it's elevated in severe patients, and when it's on the left, it's relatively less compared to the less severe patients. One thing that immediately came out, and the upper quadrants are significant, is IL-6. IL-6 being important in the innate immune response, but also a signature that was seen in China and Italy. We replicated that finding. Interestingly, when you look at the proteins that are downregulated, we found pathways that were important for lymphopoiesis, like stem cell factor being lower, IL-7 receptor for IL-7 signaling being lower, being important for lymphopoiesis, which is especially interesting in the setting of lymphopenia, observed during severe COVID. We also found molecules of proteins that were important in the kallikrein-kinin pathway, and these are basically inhibitors or neutralizers of the kinins like DPP4 or like serpin A12, a inhibitor of the plasma kallikrein. So there was a hint that the plasma kallikrein inhibition was lower, meaning that it might be elevated and activated. So from these pathway analysis of different proteins, we have three major pathways that we think are important. One is the innate immune pathway, the IL-1, IL-6, being important for chemokine cell infiltration, neutrophil influx, coming from macrophages, monocytes. On the other hand, the adaptive immune system being lower, there is a lymphopenia, and we do have some ideas of findings that basically lymphopoiesis itself is inhibited with downregulation of SCF and IL-7 receptor. And then there is the complement, which has an interaction with kallikrein-kinin system, but also itself the kallikrein-kinin system being dysregulated, activating B1, B2 receptors, which are the bradykinin receptors, which might play a role in the local lung edema. Now, if we think about innate immune activation, we think about macrophages, and we know a syndrome, macrophage activation syndrome, that is playing a role in severe sepsis or in severe influenza, and we were expecting to see this also as a problem in COVID, and it does play a role, but it's some different level. Important to explain is the autoinflammatory loop that is associated with the pathogenesis of this syndrome. So MAS, or macrophage activation syndrome, in a cytokine storm is a component, And here we focus on the innate immune cells. A pathogen is recognized here, coronavirus, by pattern recognition receptors and leads to pro-IL-1 beta production, which is the pro form of the cytokine IL-1, and it's not active, not bioactive. This needs to be processed to become bioactive, and it's done by a complex that will activate enzymes, caspase 1, that can cleave then IL-1, and it's then secreted. Once monocyte macrophages start producing IL-1 beta, the consequence will be there is induction of IL-6, there is induction of neutrophils, influx of neutrophils, and there will be fever. So that's a typically IL-1 driven response. What's important to mention is that IL-1 can induce itself. So there can be a loop where the own cell is producing its own substrate and is keeping this pathway intact for multiplication of neutrophil recruitment and more IL-6 and driving innate immunity. So it's very important to control this loop, and it can be done at several places. It can be done at the receptor by IL-1 receptor antagonists, by the own body. It can be done at the level of the inflammasome over inhibitors or secretion, but it has to be kept in check. Now, in coronavirus, there are two additional things that come into this loop. One, there is tissue damage, which will release IL-1 alpha, that together, like IL-1 beta, can induce this pathway and can persist the loop. Also, there will be dying cells around, like apoptotic cells or necroptotic cells, that can be phagocytosed by those macrophages, innate immune cells, and can also trigger pathways leading to the exact same loop. So there are some enforcing pathways during corona that can keep this loop sustained. And if you have some genetic background or other reasons that you cannot put the brake on this loop, you will have a component of an autoinflammatory loop leading to more inflammatory responses at the innate level. Now, importantly, not only IL-1 is processed in this way, there is constitutively in the cell a lot of pro-IL-18, and that will also be processed by the inflammasome, leading to IL-18 secretion, which in turn will induce interferon gamma in the adaptive immune cells, which can then cross-regulate and cross-talk with innate immune cells. And this can also be an interplay between innate and adaptive, leading to the classical cytokine storm syndrome. How do we keep this loop in check when it's not during pathogenesis? We have several options. We can choose the recombinant IL-1 receptor antagonist, which is anakinra, which is very elegant because it blocks both IL-1 alpha and IL-1 beta, as you can see. Second, we could target the inflammasome, which will then drive less of the IL-18, IL-1, and IL-6 all on the right side altogether, because it inhibits both pro-IL-18 and pro-IL-1 processing. It can be targeted at a level much more further away, like IL-6, or at the level of IL-18 or interferon gamma, basically the crosstalk between the adaptive immune system and innate immune system that you then target. So this is something that we know in sepsis, this is something that we know in influenza, but what about COVID? Because it has a component, but COVID has more features. We have learned that it gives pulmonary edema, there are unique. One of the things that can play a role is the kallikrein-kinin system in COVID-19. Here we have a summary of that system, and above there is an enzyme called kallikrein. That's a set of enzymes. One enzyme is present in the plasma, and several are present in the tissue, like 15. And there are also tissue kallikrein enzymes in the lung. Now, what do? Those enzymes, they process a preformed protein called high molecular weight kininogen or low molecular weight kininogen. They are both produced by the liver, but depending on whether it's in the plasma or in the tissue, it will be processed into a form of a bradykinin. So the well-known form of normal bradykinin being in the plasma present is coming from the high molecular weight kininogen produced by the liver. And this bradykinin has to be neutralized and kept in check by the enzyme ACE. If it's not, and you have, for example, an ACE inhibitor and the bradykinin becomes too high, it can activate the B2 receptor and lead to local angioedema. Not specifically in the lung, but local angioedema. Now, in the setting of COVID, there is something else. There is a further downstream processing of those bradykinins, which can act via B2 receptor, by enzymes like kinase 1, CPM, etc. And they will produce kinins that are basically important for activation of another bradykinin receptor, the B1 receptor. And this receptor is normally not expressed, in contrast to B2 receptor, normally not expressed in a constitutive way. Only when there is inflammation present, it will be upregulated, and then it can be signaling, leading to leakage of that endothelial at that place. And ACE2 is exactly needed to keep those ligands in check. It neutralizes basically the B1 receptor ligands. Now, how would that fit in the theory? With COVID, there has to be a setting where ACE2 is expressed at the alveolus, so at the pneumocytes type 2, and there is evidence that during COVID there is ACE2 expression. And there is a bradykinin receptor type 2 in the endothelial cell. So now, in a stage where there is a viral infection with COVID, it will get into the alveolus, will use the ACE2 receptor on those pneumocytes to enter the cell, and because of this, it will lower the ACE2 expression and thus the function of ACE2, which we do know already from SARS literature that is happening. Because of the ACE2 deficiency that is locally present, there won't be a capacity to neutralize those B1 receptors that are formed because the low molecular weight kininogen produced by the liver, which is there in the plasma, and the activated tissue kallikrein that's producing those B1 receptor ligands is not controlled by ACE2 because it's not present. Because of this, there will be a leakage, bradykinin-driven, kinin-driven endovascular leakage, and this will set a extra vicious cycle that will then turn on the plasma kallikrein-kinin system. Those B1 receptors are present because there are macrophages, there's mild inflammation, and cytokines like IL-1 can upregulate B1 receptor. Once this plasma leakage is happening, it can then activate the plasma kallikrein system, which is very potent in producing a lot of the B2 receptor ligands, like lis-bradykinin or bradykinin. Basically bradykinin because that comes from the plasma. The tissue will further produce also lis-bradykinin, and those B2 receptor ligands then add on to the plasma leakage. There is more inflammation, there will be more B1 receptor ligands, and we do think that this is not kept in check and will continue to be present as long as the virus is there and there is no break on this system, and as there is an ACE2 deficiency, it will maintain. Now, what we did is we started looking if we could find evidence of an activated kallikrein-kinin system. We measured the fragments of the plasma kallikrein in the plasma of those patients, and we found significantly elevated fragments, saying that our hypothesis was also supported by, first, the signatures that we found of the inhibitors that were lower, and second, there was a plasma kallikrein activation. So we said, if this is true, we might help those patients, preventing their angioedema in the lung with blocking bradykinin receptors, and there are blockers known and on the market for bradykinin receptor type 2, which are used for hereditary angioedema. So when we treated 10 patients with icatibant, the drug that blocks bradykinin 2 receptor, we found that there was a significant reduction in the oxygen status need of those patients. You can see on the y-axis the oxygen need in liters per minute, and most of them, they were ill, like they had 3 to 7 liters needed, And within 24 hours, they had a clear reduction, while if we match those to historic controls, over time, we never saw this really short interval where there was a significant drop like this in the patients, what we observed in the patient with icatibant. If you visualize this differently, we here have the status when they start in the lighter color of the oxygen need that they had, and then the drop, the maximum drop in the amount of hours that was there, and you can see 14 hours. One patient took 38 hours, but most of them 22 hours, 15 hours, 24 hours. And even patient 9, for example, in 10 hours came from 5 liters to 0. Meaning that we significantly observed a drop in oxygen need due to icatibant treatment. And when we matched that to controls, there were only a few that spontaneously had a few oxygen liters lower, but never this significant drop of oxygen need due to the ones that we observe in icatibant. Important is to mention that there were a few patients that had a resurgence because the drug doesn't last long. It has a very short half-life. And here you can see, for example, coming a patient from 7 liters to 1 within a very fast period, 24 hours, that there was a resurgence of the oxygen need following the normal slower or lowering that would have followed. Similar in patient 5. So this is strengthening us that when the icatibant was present, there was really something we're doing on the hypoxia, and when it was not present anymore, the patients came to their normal status. So because the drug doesn't last long, we need to do something different. So we're now targeting the plasma kallikrein itself with a drug called lanadelumab, which we are trying to give intravenously, so it works fast and it works long. It's a monoclonal antibody against plasma kallikrein, and we are setting up that trial. Now, who and when to treat? One of the first questions that you have to ask, are there very many different endotypes, for example? Are all people going through the same stages of the disease? So what we did here is we took all the analysis from the proteins that we measured, and we put them into a analysis where the computer tells us if they cluster or not. And as you can see on the y-axis, all the blue patients are patients admitted to the ward, and all the reds, patients in red, are admitted to the ICU. And the computer could very perfectly match those according to the levels of the proteins, and if they were up or down, they all had the same signature, and there was not a clear difference in endotype. And then when you look to the proteins that were up in the ward and that went down in the more severe, they all had the same gliding scale from proteins moving up to down when they become more severe, or they were low on the ward and they got upregulated in the more severe state of COVID, like ferritin and D-dimers, CRP, IL-6. So this means that basically everybody glides through the same pattern, not very significant for a specific endotype. So when you target, and these are patients that these are time points when they entered the hospital, so very early in their COVID, not talking about later stages in the ICU after 2 or 3 weeks. There are clear differences between patients. But here, if you look at the beginning, you can see that most of the patients have the same scale and problems, which we do think are the virus being present, having local pulmonary lung edema, plus an innate immune response that's on, and a lymphopoiesis that is downregulated. Now, how would that translate into a treatment strategy and timing? Obviously, you can do interventions before disease like vaccination, prophylactic antivirals. But when you start the disease, you need a good antiviral. And when you progress into problems with pulmonary edema, you need to target that initial mechanism where we think that ACE2 directly by the virus contributes to that local status of more fluid, and that you target the kallikrein-kinin system. If you then progress during the disease, the first signs and signatures that we kept and which is all over all the observational studies, is that your innate immune system with an IL-6 signature is activated and that there is a lymphopenia. So we do think that you need to target the hyper-inflammatory status, and we think it would be elegant to do this with anakinra, blocking IL-1 alpha and IL-1 beta, but there is also certainly something to say for corticosteroids or maybe IL-6 targeting or complement. At least you have to do something anti-inflammatory in addition to the leakage problem. If you then don't, or if you are at a later stage and you're not getting the patient better, you run into more problems which are much more related to the endothelial status, like thromboembolic events and maybe later stage even fibrosis of the lungs, which can be very difficult to treat. Once you are in this stage, there is a lot of extra pathways crossing and crosstalking to each other, which makes it much more difficult to target. But you should do then at least anticoagulation, targeted therapy, fibrotic targeted therapy, in addition to antivirals if the virus is still present, and maybe even the kallikrein kinase system is not playing at this stage a very important role anymore. It's really, we do think at the beginning that you should target this. So we did this with a lot of people, which I would like to thank. It was very interesting to see how the COVID developed and how we learned in a very short time a lot of pathways and novel pathways that are important. And thank you for your attention.
Excellent presentation, Frank. Really interesting data and insights into a very unique aspect of COVID infections. For the audience, the Q&A is going to begin in just a moment. So just want to remind everyone again to submit questions for our panelists. Send in any question that you think is interesting. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit submit. Okay, since we have a ton of really great questions, let's get to the Q&A and answer as many of them as possible. Bear with us for just a moment as we transition into the Q&A session.